to detect mutants after creating them in a lab, for example, is to observe changes in phenotype. And hopefully the changes are significant enough that we can observe them in simple assays. So as an example of a simple assay, here's a biofilm assay in which this is the wild type, and this little purple band here is the biofilm that this organism has made on a glass tube. And then we stain the biofilm with crystal violet so you can see it better. This particular mutant here is deficient in an amidase gene, and so as you can see, the wild type produces biofilm, but this guy produces significantly more, suggesting that the loss of this particular gene, this amidase gene in C10, encourages more biofilm formation than the wild type. And in this particular mutant, you've also lost, and you've done the opposite, where you lose the biofilm. And so something about this gene seems to be a positive activator of biofilm, whereas the loss of this gene suggests that it's a repressor of biofilm, because when you take it away, biofilm goes through the roof. Another approach that we can use is replica plating. And this technique allows us to screen lots of mutants at the same time for a particular trait. And so we often use it to detect auxotrophic mutants. And I'll show you that here in this slide. Use of environmental conditions in which only the desired mutant will grow is another approach. So imagine we're looking for mutants that are less sensitive to a particular antibiotic compared to their wild type parent strain. And so we could then put the antibiotic in the medium, and then we would select for the growth of those more resistant mutants. So in this slide, you see the replica plating workflow. And so here's our starting tube of wild type E. coli, for example, and they've been exposed to a mutagen, either a chemical mutagen or it could be UV light. And so we would then plate these guys and then get a nice dense colony spread. And then we can use a replica plater like this to collect the colonies, simply just create a grid of the colonies on the velvet surface here, and then we can stamp those onto the surface of plates. The first plate here is a complete medium, so everybody should grow. It's the same medium that you see here. The second medium is a medium missing, for example, in this hypothetical case, lysine. So what we're looking for then is lysine auxotrophs. All strains grow on the complete medium, but over here, pretty much all the strains grow except for this one colony that's missing. The colony exists here, but its replica plated partner is not present. And so that mutant then, this guy here, would be our lysine auxotroph that we can grow and work with. So not surprising, microbes have all kinds of mechanisms for repairing damaged DNA rather than it just being fatal and them dying. So for one thing, they have proofreading polymerases that can correct damage or any kind of errors that they make. But another couple approaches are illustrated in these next few slides. The first one is excision repair, which can correct damage that causes distortions in the double helix. Usually the end goal is to remove the damaged portion of DNA and then refill it with the correct DNA. And so that's pictured here, and there's a couple important proteins that you can see in terms of their mechanisms of action. And so in excision repair, typically it starts with UVRA and B. These molecules, these proteins are sliding along the DNA helix in this model and looking for distortions in the DNA. This happens to be a thymine dimer, which imagine it having occurred as a result of UV exposure. Once it finds that lesion, the UVRAs will drop off, and that'll recruit UVRC, which cuts the phosphodiester backbone, flanking the thymine dimer. And so that cut then allows UVRD to come in as a helicase and strand separate these, essentially breaking the hydrogen bonding here. And that error containing fragment can be lifted away. And then something like an error fixing polymerase like DNA polymerase 1 can then fill in the gap with the correct bases. There's also a direct repair mechanism called photoreactivation, and as the name suggests, it requires light. And so the microbes we'll use an enzyme photolyase to facilitate that. And so that's pictured here. And so here's another pyrimidine or thymine dimer caused by, for example, UV. And so here's our photolyase enzyme that recognizes that distortion. And so again, this is happening when there's light, whereas this can happen usually in the dark, but if there's light, this thing can also function as well. This only happens when there's the right amount, the right kind of light, the right wavelength of light. And so the enzyme is excited by the right wavelength of light. It turns into an exonuclease in a way and cuts that out, and not cuts that out. It just breaks the abnormal bond between those two thymidines, and then restores the natural phosphodiester backbone. One of the coolest organisms is Deinococcus radiodurans. And this Deinococcus is a microbe that can handle unusually high levels of gamma radiation. And it's almost scary that this organism is so radiation tolerant. And so in this particular experiment, what you're looking at is DNA fragment size over here and time post-irradiation here. And so this control represents a Deinococcus strain that was not exposed to any radiation. And then they take the genomic DNA from the organism and cut it with some enzymes to produce the banding pattern that you see here. So then they expose Deinococcus to a certain amount of gamma radiation and let it sit at 0 time. So imagine the tube of Deinococcus gets blasted with radiation. The radiation is turned off, and then you take an aliquot of that and you do a genomic DNA extraction, and you run the DNA on a gel, and you get a smear, basically, because the DNA is just completely scrambled. You take that same tube and wait 2 hours, take another shot of that, and run that on a gel, still scrambled. 4 hours post-irradiation, still scrambled. 6 hours, still scrambled. 8 hours, still scrambled. But by 22 hours, almost a day later after exposure, the cells are not dead, believe it or not, even though their DNA is completely scrambled. They have repair mechanisms that slowly start to fix the DNA, and you start to see it again. And then after 30 hours, the DNA is basically repaired to the level of wild type. Just an incredible example of DNA repair mechanisms that allow this organism to endure something as completely destructive as gamma radiation.